Hello everyone, in this video I will talk about flecanide. The reason why I chose flecanide is because I'm interested in learning about how flecanide regulates the electrical impulse of the heart. Now this is the structure of flecanide. It contains a secondary amine, an amide group, two ether groups, a benzene ring, and 6 fluorine atoms. The melting point of flecanide is 140 degrees C, and the boiling point is 434. This is the IUPAC name of flecanide. So what is flecanide and what is it used for? In general, it regulates the abnormal heartbeats, and it belongs to the IC class of antiarrhythmic drugs. Okay, so the IC class of antiarrhythmic drugs bind and block sodium channel in the heart. So here's a short video that explains how the electrical signals of the heart spread.
The heart's electrical system includes three important structures: the SA node, the AV node, and the His-Purkinje system. They trigger and carry electrical signals through your heart to make it contract and relax. The SA node is a bundle of special cells in your upper right atrium. These cells create the electrical signals that trigger your heartbeat. The SA node is sometimes called your heart's natural pacemaker. Electrical signals triggered by the SA node travel through the atria to the AV node. The AV node is a bundle of special cells between your heart's upper and lower chambers. The AV node can be thought of as an electrical bridge. It allows electrical signals to travel from the atria to the ventricles. The His-Purkinje system is in your heart's ventricles. Electricity travels through the His-Purkinje system to make your ventricles contract.
This picture here depicts how the electrical impulse spreads within the heart. Flecanide is responsible for slowing down the conduction of the electrical impulse. It has a great effect on the His-Purkinje system, which we just learned about, and it has a great effect on the left ventricle, which leads to a decrease in the ejection fraction. Now, the ejection fraction is basically how much blood the left ventricle produces with each pump. Now, flecanide is fully synthetic. And before synthesizing flecanide, 3 other drugs were commonly used in treating cardiac dysrhythmias. One of those 3 drugs is called lidocaine. Here's the structure of lidocaine. It is similar in structure to flecanide. It has an amide group, a benzene ring, and it also has a tertiary amine. Now, chemists had the idea of introducing a CF3 group to the antidysrhythmic drugs. The compounds that were used when conducting testing were fluorinated derivatives of local anesthetic and anti-dysrhythmic drugs. Now, 2 synthetic routes were used to produce flecanide. The first assay tested the ability of the compounds to avert the ventricular fibrillation. So the second route involved fluorinated, it of course involved the same compounds, which are fluorinated derivatives of local anesthetic and antidysrhythmic drugs. It involved testing dogs in a conventional way. Now, Flecaninde was approved in 1984. Here's the synthesis of Flecaninde. Now, when I looked at it and tried to analyze it, these H's, now there's two alcohol groups here and one carboxylic acid group. Each H was replaced by the CF3 groups, as we can see here. But then they used different compounds, and both compounds led to the synthesis of Flecaninde. Okay, now that we understand how Flecaninde affects the heart in general and how it regulates the electrical impulse, we need to understand how Flecaninde works on a biochemical level. Okay, here's the RCSB website. This is the protein code that I chose to study. And this is the origin of Flecaninde, which basically originated from these two organisms. So as we can see here, this is the sodium channel. And Flecaninde works by blocking that channel in the heart. Now, Flecaninde waits for the channel to open, then it attacks it and it blocks it. Now, this article was written by these authors here. This picture here depicts how Flecaninde waits for the channel. So basically, here's the Flecanide molecules, and here's the gate of the channel. So Flecanide is waiting for the channel to open, which means it's waiting for the channel to achieve its inactivated state. So this is the channel after Flecanide attacks it, and here's the Flecanide when it's trapped within the inactivated channel, basically. Okay, this picture depicts a cell membrane. This is the heads part, which is hydrophilic, and this is the tails part, which is hydrophobic. Okay, this is a 3D model of a cell membrane. Now this is, as we can see here, this is the heads part, this is the tails part. Now, because we know that like attracts like, we should assume that sodium channel is hydrophobic as well. Because if it weren't hydrophobic, then that would lead to a repulsion between the channel and the hydrophobic part of the cell membrane, which is the tails part. Okay, here's a pocket version of the sodium channel. I color-coded the amino acids. The green part represents the hydrophobic amino acids. The yellow part represents the polar amino acids, and the blue part here represents the positively charged amino acids. Now this proves my hypothesis about the sodium channel being mostly hydrophobic. And if it weren't hydrophobic, then, like I said, it would lead to a repulsion between the sodium channel and between the cell membrane. Okay, here's a better picture of the sodium channel and flecanide. It shows how flecanide clearly resides inside the sodium channel, and it looks like a cavity, like flecanide resides inside a cavity. Okay, so when I started analyzing flecanide, the interactions between flecanide and the sodium channel, the first thing that I thought of was hydrogen bonding. And the reason why I thought of it, because flecanide has 6 fluorine atoms, which could potentially react with the sodium channel via hydrogen bonding. However, it turned out that I was mistaken. There isn't really any hydrogen bondings between flecanide and the sodium channel. So I eliminated that option immediately. Now, the second option that I had was dipole-dipole interactions. However, I couldn't find any proof for dipole-dipole interactions, and also it did not make any sense that there was dipole-dipole interactions between flecanide and the sodium channel. As a result, I also eliminated that option. Now, because I know that the channel is mostly hydrophobic, I immediately thought of London dispersion forces, and it does make sense because London dispersion forces depends on how polarizable the atom is, and it also depends on the contact surface. So the bigger the surface, the stronger London dispersion forces is, right? Okay, here's a short video that explains what London dispersion forces are. So as we can see here, those are 2 non-polar compounds. Now, when they get close to each other, one compound will develop a partial negative end, and the other will develop a partial positive end. Those are instantaneous dipoles, and as we saw in the video, they fade quickly. Now I want to talk about another type of interactions, which is called pi stacking. Now this is a benzene ring, and we can see that this part is negatively charged while this part here is positively charged. So it turns out that aromatic rings, like benzene rings, interact with each other, and it's, like I said, it's called pi stacking. Now there's 3 types of interactions that happen between benzene rings. There is the face-to-face interaction, there's the parallel staggered, and then there's the T-shaped interaction. Remember when I mentioned that flecanide has a benzene ring? Well, the benzene ring of flecanide does react with the benzene ring of phenylalanine. And the type of interaction is the T-shaped interaction, because as we can see, it is the benzene ring is kind of perpendicular to the benzene ring of flecanide. And so we can imagine that this end here is positively charged, while this part here is negatively charged. Now, there is another type of interactions that's responsible for binding flecainide to the sodium channel. Now, according to this article that was written by these authors here, that type of interactions called cation pi interactions with phenylalanine. Okay, so what is cation pi interaction? In general, it's an interaction between electron cloud of an aromatic compound and a positively charged cation. And this is the phenylalanine amino acid that was mentioned in the article. So this benzene ring has an electron cloud, a polarizable electron cloud that attracts that cation. We also should understand that this type of interactions happen with aromatic compounds like benzene rings. Okay, now if we go back to this article, it says here that it is possible that the positive charge of flecainide could similarly interact. And in this article here, it says that at neutral pH, the amino acid group is protonated. Now we'll go back to chimera and see how that is possible. So I color-coded the phenylalanine amino acids. And if we take a look at flecainide, okay, it contains an amino acid here, and at a neutral pH level, this amino acid would be protonated, and this would make sense because here we have a benzene ring of phenylalanine, which would attract this positively charged nitrogen here. Now, it turns out that there is another type of interactions that bind flecainide to the sodium channel. It's basically an interaction between a pi pyridine ring with phenylalanine, and it's specifically the hydrophobic part of pi pyridine ring that interacts with phenylalanine. This is what a pi pyridine ring looks like. And here's the pi pyridine ring of flecainide. It's this one here. And this is the hydrophobic edge of the pi pyridine ring. Okay, so we can see, so this is a pi pyridine ring of flecainide, and this is the phenylalanine that it's reacting with, and this is the hydrophobic edge of pi pyridine that's reacting with phenylalanine. So this is one of the interactions that basically responsible for binding flecainide to the sodium channel. Here are the references. In this video, I talked about general information about flecainide and how it regulates the electrical impulse from the organ level. Then I talked about how it reacts with amino acids of the sodium channel. Hope you enjoyed it, and thank you for watching.